I'm Thomas Riffelmacher, but everybody calls me Tom. I'm an instructor here at Jolla Institute. More recently, I've also been directing the Immunometabolism core facility. So here we are in the room with our favorite instrument, the Seahorse Metabolic Flux Analyzer. And this is the XF-96 version of the model, which basically means that we can actually measure 96 wells of cells, of immune cells, at a single time. And so there's other versions that only have 24 or 8 wells, but here specifically I think we really wanted to have the capability of looking at many different immune cells at the same time, in real time. And so many of us study T cells or myeloid cells or different types of immune cells. And we put a few 100,000 cells in there. And then in addition to the plate here, we have this probe sensors. So here on the bottom of that, we have a little membrane with biosensors on each one of these probe ports. And these sensors basically go into that plate like so, and then they create a micro chamber at the bottom. So this chamber is about 7 microns high. It's quite small, and it's sealed off, meaning that in this micro chamber is where the magic happens. And what's the magic? What this instrument is doing is it's basically measuring in real time the consumption of oxygen and the acidification of the medium. So what does that mean? And here you can actually see those two readouts. OCR, that's for oxygen consumption rate, and then ECAR or ECAR, that's the extracellular acidification rate. So those two measurements are telling us about the metabolic state of these cells. Oxygen consumption is being measured by a sensor on that cartridge that detects changes in oxygen levels. And then there's a second sensor on the same membrane that basically it's a sophisticated pH meter. It basically measures protons, change in protons in real time. And so these two readouts are happening in this machine in real time. And so what they tell us is how much oxygen is being consumed, and oxygen consumption, of course, occurs in the mitochondria as the cells respire. They're using mitochondrial oxidative phosphorylation, and that uses up oxygen. And then the extracellular acidification rate, that's measuring how much glucose is being consumed, and during that process protons are being released into the medium, so the pH drops very, very slightly. And so each proton that is being released basically tells us that glucose is being consumed during glycolysis. And so as these two readouts then occur over time, we have the option to also inject specific chemicals through these ports. And the compounds that we use in this particular assay are 3 compounds. The first one is oligomycin. So what it does is it basically inhibits the mitochondrial energy production, the ATP synthase. And then what happens, as you would expect, is this oxygen consumption goes down. And this change in oxygen consumption then basically tells us how much in this cell has oxygen being consumed to generate ATP in the mitochondria. And because we're taking that away, glycolysis is compensating to some extent. You can actually see that simultaneously the extracellular acidification rate then goes higher. And then what we do is we use this FCCP. It's a mitochondrial uncoupler. And so basically it causes the electron transport chain to run at its maximum. So the mitochondria are just being asked to go full speed. And what we see is, not surprisingly, the oxygen consumption shoots upward, and then this level where it steadies out is the maximal respiratory capacity that these cells can do. And then finally, the third injection here is a drug called rotenone, and we actually also add antimycin A. And this basically shuts the whole mitochondrial pathway completely down, and you see the graph drop way down to the bottom. And this is, in a way, it's a control, and it tells us how much background of oxygen consumption is occurring that's not related to the mitochondrial energy production. And so what this allows us is basically to create energy maps of our immune cells. So we can see here, for example, one example of an energy map. You see on the x-axis here is the acidification rate, and then on the y-axis you have the oxygen consumption. So if both of these are really high, this is an energetic metabolic state. And one example are effector T cells that usually when they're activated and producing cytokines, they'll be way up here. They'll be actively using both of these pathways. Whereas on the opposing side here we have quiescent cells. So for example, some stem cells and memory cells can have a lot more quiescent metabolism. Then there's also the aerobic and the glycolytic corner of this. So in the aerobic corner we would have cells that prefer using oxygen over glucose consumption, and in the glycolytic rate of corner, of course, these are cells that don't use so much oxygen but are highly glycolytic. I think the power of this instrument is that it gives us this insight into how these metabolic pathways are regulated and active in real time. And I think the revolution has been that this is kind of correlating with the functions of these cells. So I think previously people thought that a lot of these metabolic pathways are kind of a housekeeping or kind of a background thing that's just going on and not so important. I think more and more we are understanding that, particularly in the case of immune cells, particularly in the case of T cells, which are the cells that I'm studying, oftentimes we see that certain metabolic states, not only do they correlate with certain immune states, with certain effector functions, but we can meddle with, we can impact or tune these metabolic states, and then that will impact what type of T cell responses, what type of immune responses these cells will prefer or are most suited to carry out. And so that's, I think, really interesting and therapeutically, of course, has potential to give us a handle or a lever that we can use to change or modulate the immune responses in therapeutic settings. So this is where the plate goes in, and so here we have the plate with the sensor cartridge, and then it sits on top of here and then disappears. And so this box is actually kept at exactly 37 degrees Celsius. So this is the temperature that we also use to incubate our cells, and where they'll be active and will actually be measuring their metabolism in this setting. We actually have this BSL-2 plus setup here. So that means we can use bacteria, we can use virus-infected cells. If we want to actually measure cells that are infected, we can do that here thanks to this setup, and we can actually measure the impact of an infection on the metabolism of these cells in real time. A lot of the other researchers here, when they first come to me, they've had some preliminary data from, for example, their transcriptomic data sets. We have a fantastic RNA sequencing core here, so many of us have this single cell transcriptomic data of their immune cells in different settings. And of course, in these data, you can already look at metabolic pathway activity on a gene level, transcriptional level. And sometimes these pathways are regulated at that level, and you will already see some changes, and then people are wondering, well, does it actually translate into real differences of metabolic pathway activity in these cells in real time? And so, yeah, that's when people usually come to me, and we figure out ways of looking at the metabolism in their immune cells. And there's a couple of different things we can do to look at the immune cells, and a lot of it depends on how many cells are available. If it's super rare cells, this is not the perfect instrument. So this instrument actually uses, let's say, between 100 and 300,000 cells per well. The Seahorse is measuring real-time flux of these two readouts, right? It measures acidification rate, so a proxy for glycolysis, and oxygen consumption, a proxy for mitochondrial oxidative phosphorylation. If we want to dig deeper, we have the option to look at metabolomic data sets. So this is what we've been doing more recently, and we've actually had, I think, 3 or 4 projects now at the Institute where we looked at metabolomic data sets. So we generated metabolomic data sets by carbon tracing. How that works is basically we have a molecule that's isotope labeled. So in some of the work, for example, we used a glucose, but instead of the carbon-12 isotope, which is the regular isotope for glucose and for carbon, we have a C13 labeled glucose. So the carbon is one tiny bit heavier, and this is something we can measure by mass spectrometry. And so what we do in these assays is we culture our isolated immune cells with this heavy glucose, and over time the cells are incorporating this glucose into their metabolic pathways. And then we take those cells and we extract all the metabolites, and then we send it to mass spectrometry for analysis, and then we can actually see how much of that heavy C13 labeled carbon has been taken up by the cell, went from glucose to glucose 6-phosphate, and then from glucose 6-phosphate down the glycolysis pathway into each of the metabolites. So you get this incorporation down through the pathway, and you can follow it, and at the end of glycolysis you have acetyl-CoA, and that acetyl-CoA then it's incorporated into the TCA cycle, the Krebs cycle. And again, we can follow all the individual metabolites of the Krebs cycle, and we can see how much of that heavy carbon has gone into citrate, has gone into isocitrate, has gone into alpha-hydroxyglutarate, and so on, malate, and so on. And then we can construct basically a map of all these metabolic pathways and these individual metabolites and follow how they're actually being used and routed into the pathways that are active in the cell. And so that gives us a lot of detailed insight into what is really going on at a molecular level in this cell. Here at LJI, a lot of us are interested in the metabolic state of immune cells. And in many cases, we have mouse models of diseases, and oftentimes we also have human cells. And so that's usually that's the two most common starting materials. And oftentimes the researchers already have isolated the immune cells they're interested in. Oftentimes they already have transcriptomic data sets. So from RNA sequencing, we have the gene expression of all the genes in their cells. And from this data we can already look at some of the metabolic pathways. And sometimes, but not always, that correlates with the actual function or activity of these pathways. But what we then do basically is we ask how in real time these pathways are active. And one of the first assays I like to do is to look at consumption of metabolites by what I call flow metabolic assay. That's something that I've been doing a lot in my previous work in University of Oxford during my PhD. I was doing a lot of these assays, and I like this because it gives us an overview, and it's relatively simple to do, and it has single cell resolution. So how these work is we give labeled metabolites to either the animal or the cells in culture. And this can be a fluorescently tagged glucose, or it can be a fluorescently tagged fatty acid, a glutamine, etc. And then we allow this to be incorporated into the, taken up by the cells and incorporated into the pathways. And we measure, well, how much of that nutrient has been taken up. And because it's fluorescently tagged, we can do this by flow cytometry and just quantify the fluorescent signal, and that will directly correlate with the amount of nutrient that has been taken up. We can, in addition, do these assays of organelle homeostasis. So these are basically assays where we quantify the number of mitochondria, the activity of the mitochondria, the lipid stores, and other metabolic organelles. Again, all of this can be done on single cell level and by flow cytometry or flow metabolic assays. And then we can create maps of energetic phenotypes, etc. And this really is great to get an overview of sort of the states that are present. But then we can do more detailed assays. So this is where the Seahorse bioanalyzer comes in, measuring real-time flux through these pathways. And we've done, together with the electron microscopy core at UCSD, electron microscopy, actually looking at the mitochondria, looking at the structure of the cristae, seeing if there's changes in the morphology, the elongation, etc. And these things tell us about activity as well. And then finally, and I think recently that's become quite attractive to several people here, is doing carbon tracing metabolomics, where we really measure then on sort of the finest level of detail, I guess, the number of metabolites that are being incorporated and where they're going in the cell. For my own work, I've been looking at these mucosal associated invariant T cells. They're this unconventional T cell type that's very abundant in liver and lung and in mucosal tissues. And what we found is that there is two kind of flavors of these cells, and we have a live vaccine strain. It's actually a bacterial vaccine strain that leads to a massive expansion of these T cells. But they not only expand, they also adapt. So they become sort of like we call it memory-like, because it's a long-term change in their phenotype, and they're more responsive to a secondary insult, or to a secondary pathogen challenge. And they had these two flavors that were incredibly different for their metabolism. We have one that is, we call them MATE1-like, and they have this Th1 cytokine phenotype, and these ones have they're much more dependent on glycolysis, and they have this ready-to-respond state where they have actually similar numbers of mitochondria than the other flavor, but they're very quiescent and they sort of shut down. And then that other flavor is these MATE17-like cells, so they produce a cytokine called IL-17, and they're very good at fighting extracellular bacteria. And they had a completely different metabolic phenotype. They had really high oxygen consumption, and they had high levels of active mitochondria that were fed by fatty acids. Whereas these MATE1-likes, they didn't consume fatty acids at all. And the seahorse was actually really important in defining these two metabolic states. And when we saw the first sets of data, we were just really impressed by the large differences in. It's a very well-defined cell type, and yet you have these two flavors that have completely different metabolic phenotypes. And then the cool thing is that we could demonstrate that by interfering or modulating these metabolic pathways, we were actually able to change the functional phenotypes of what type of responses we get. So we could interfere with the oxygen sensing, and that would reduce the mitochondrial activity, and then we actually got more of those MATE cells that are MATE1-like, that don't use the mitochondria, and these ones were very good at fighting, for example, a viral infection. So we could demonstrate that they were better to protect from an influenza challenge.